Okay, greetings everyone. In this video, beforehand, my name is Eva, and in this video, me and my colleagues Farhan and Gaiska, we will be talking about human and animal cells in pharmacy and human diseases. Before we start the topic, we will cover what are human and animal cells first. So human and animal cells are eukaryotic cells. And the definition of eukaryotic cells are cells that contain membrane-enclosed organelles. Now, how to differentiate eukaryotic cells with prokaryotic cells is that prokaryotic cells lack membrane-enclosed organelles. Both eukaryotic cells and prokaryotic cells contain ribosomes, but they have different structures and compositions. So humans and animals are complex organisms with multiple organs that carry out different functions.
So as Eva has said, animals and humans are both complex organisms, so they need different cells that can fulfill the functions of different organs. So today we will be talking about the nervous system, and in particular the neurons. So the nervous system is often referred to the command center of the body. It is responsible for the transmission and receiving of nerve impulses via neurons, and it can be further classified into central nervous system consisting of the brain and spinal cord, as you can see in the diagram, and the peripheral nervous system, which is all the neurons that transfer information into and out of the central nervous system. And apart from the neurons, there are structures known as neuroglia, which are supporting cells that help to maintain the physical structure of the nervous tissue. Since there are so many transmission signals that come from all the neurons in our body, there is a need for a brain or a ganglia, which interprets information transmitted by these neurons.
As Gaiska said, today we're going to talk more about neurons in our nervous system. Now, as we look closer, neurons have structure and functions. Most of the organelles in the neurons are located in the cell body. From the cell body protrude extensions known as dendrites that receive signals from other neurons. They have a single axon that transmits the signal to other cells. Axons are typically longer than dendrites. At each end of the axon, information is transmitted to another cell in a junction known as a synapse. At this junction, chemical messengers known as neurotransmitters are used to pass the information from the transmitting neuron to the receiving cell. Aside from that, there are also the Schwann cell, also called neurulima cell, and it's any of the cells in the peripheral nervous system that produces the myelin sheath around the neuronal axons. Then there's also the node of Ranvier, a periodic gap in the insulating sheath of myelin on the axon of certain neurons that serves to facilitate the rapid conduction of nerve impulses. And finally, there's the myelin sheath itself, which allows electrical impulses to transmit quickly and efficiently along the nerve cells. Neurons then can be classified in terms of the type of transmission signals they receive and how they handle such signs. They are sensory neurons. They receive and transmit information regarding internal stimuli. In this case, is blood pressure, blood sugar level, and also external stimuli, in which smell, light, or touch. Then there's interneurons that connect the neurons in the brain or ganglia and are responsible for the interpretation of the sensory input. And finally, the motor neurons that transmit signals to effector cells or tissues or even organs.
Okay, so next we will talk about how neurons work. So in other words, the transmission of messages or the action potentials. So just like any other cells, neurons contain ion channels. And when a neuron is at rest or no action potentials are occurring, the concentration of potassium ions inside the cell is 140 millimolars, while outside of the cell, the concentration would be 5 millimolars. This chemical concentration gradient causes the net outflow of potassium ions. Now, these resting neurons will then have a lot of open potassium ion channels, but a few open sodium ion channels. And this is what you call the resting potential. Next is the action potential. So neurons have gated ion channels where it opens or closes in response to the stimuli. This results the rapid inflow or outflow of the ions inside neuron that either causes depolarization, which means there's less negative concentration inside the cell compared to outside of the cell, or hyperpolarization, which means there are more negative charges inside the cell compared to outside of the cell. So if a neuron depolarizes, an action potential can be generated, and this causes a threshold of negative 55 millivolts should be reached. And this magnitude action is independently triggering a stimulus. Action potentials can either occur fully or not at all, depending if it reaches the threshold or not. Action potentials also transmitted from one neuron to another through synapse, which was explained before by Farhan.
So as we have explained, you can all see how important neurons are in our body. So damage to these neurons can have deadly consequences. And epilepsy is one disease that is a result of that. So what is epilepsy? Epilepsy is abnormal circuit activity in the brain that causes frequent seizures, while seizures are defined as bursts of electrical activity in the brain. So as Eva explained, neurons carry information in the form of action potentials, which are basically electrical energy. So if there's too much electrical activity in the brain, a person can suffer from a seizure. So a person is said to be suffering from epilepsy if they experience any one of the following conditions: at least 2 seizures, either unprovoked or reflexive, occurring over 24 hours apart, 1 unprovoked or reflexive seizure, and are likely to experience another one after 2 unprovoked seizures, or a diagnosis of epilepsy syndrome. So as you can see from these pictures, the one on the very top right here is a comparison between a normal brain activity and an epileptic brain activity. In the epileptic brain activity, you can see that the activity is not very spread out and is very concentrated in certain parts, and certain parts don't even have activity at all. And in the picture below, you can see that there is a specific seizure hotspot, that if there is too much brain activity there, the person will suffer from a seizure.
As Gaiska has explained, the causes. Sorry, as Gaiska has explained, the comparison between a normal brain and a brain that has epilepsy, and also the definition of epilepsy, I'm going to continue with the causes. Now, the causes of epilepsy are often associated with structural brain abnormalities that are genetic or congenital disorders, such as chromosomal disorders, inherited metabolic and mitochondrial disorders, malformations, or of cortical development. Now, this also can be caused by existing conditions such as cerebral palsy, hippocampal sclerosis, cerebrovascular disease, neoplasms, head trauma, neurodegenerative disease, or cerebral infections.
Now, there is no cure for epilepsy currently, but physicians and healthcare professionals will try to stop or prevent seizures from happening. Now, patients will be prescribed with anti-epileptic drugs or AEDs. Now, a common drug that will be prescribed would be sodium valproate. Now, this drug stabilizes the excessive electrical activity in the brain, increases the activity of nerve calming agents in the brain, or CANA, prevents buildup of electrical signals in the brain, but it also can cause complications in pregnant women. women. Now, there would be side effects to this drug. Some would be stomach pains, diarrhea, dry or sore mouth, tremors, feeling tired or sleepy or fatigue, headache, weight gain, thinning hair, the thinning of hair, or irregular period.
Another example of diseases that are caused by damage to nerves is multiple sclerosis. So this is different from epilepsy in the way that it is an autoimmune disease, which means the body attacks itself. So it is an autoimmune disease that attacks the protective myelin sheath. The damaged nerve fibers are around the brain and spinal cord or the central nervous system. Since the myelin sheath is needed to insulate the axon so that the action potential can be transferred or transmitted correctly, it can cause communication problems between the brain and the rest of the body. So symptoms can affect the movement and vision of patients suffering from multiple sclerosis, such as numbness or weakness in one or more limbs, electric shock sensations with certain neck movements, tremor, partial or complete loss of vision, prolonged double vision, blurry vision, slurred speech, fatigue, dizziness, tingling or pain in parts of your body.
Now, as for the drug treatments for multiple sclerosis, unfortunately, there is currently no cure. But instead, there are treatments typically focused on speeding recovery from attacks or slowing the progression of the disease. Even the severity sometimes, people have such mild symptoms that no treatment is necessary. One of the drugs in the treatment is a fingolimod, is a drug used to treat this disease and to reduce its symptoms. It affects the sphingosine 1 phosphate receptor that create T and B cells. Also, T and B cells normally kill viruses or bacteria, but in multiple sclerosis, they damage the myelin sheath instead. Also prevents the T and B cells from leaving the lymph nodes where they are made.
That is all we have from this topic. If you have any questions, please write them in the comment section. Thank you.
Thank you.